Our first talk will be given by Professor Xuejin Chen. So Xuejin Chen is currently a professor at the University of Science and Technology of China. She worked as a postdoctoral scholar at Yale University from 2008 to 2010. She has been a well-recognized expert in 3D modeling, geometry processing, and biomedical image analysis. And today, she will be talking about 3D reconstruction and morphological analysis for neurons from large-scale microscopy images. So please, let's welcome Professor Xuejin Chen. And now I will stop sharing my screen. And it's yours now, Xuejin.
Okay, can you see my screen?
Yes.
So, now you can see my screen, right? You can see my slides.
Yes, yes. And I can also see your cursor, your mouse.
So, thank you. Thanks, Professor Zhang, for introduction. And it's my great honor to introduce our work. The title of my talk is 3D reconstruction and morphological analysis for neurons from large-scale microscopic images. Actually, when I was invited for this session, I struggled a lot to figure out, a proper topic for the audience in computer graphics. And I decided to talk about our work on neuron reconstruction and morphological analysis. It seems that it's very different for the research in computer graphics, but actually all the techniques and methods we use all from 3D vision and 3D graphics community. So I wish I could provide a different perspective for the audience in CG community to see how we can apply the CG techniques in different areas. Well, because of the fast development of various data acquisition methods, we can capture color images and 3D point clouds and volumetric data. And we have witnessed the significant success of 3D modeling technique in different scenario applications. For example, the virtual reality, scene understanding, and the human-computer interactions. And actually, because of the great success of the 3D modeling techniques, it has also been widely applied in biomedical areas. And today I will talk about our work, in particularly for neuroscience. And we know that a lot of countries have launched the Human Brain Project, which aims to investigate the multi-level organization of the human brain at different spatial temporal scales. And because of the fast development of the data acquisition and data computing and the storage, I think we get a huge data explosion for biomedical areas. And for scientists in neuroscience, they spent a lot of time to build a human brain atlas from this in multiple scales in space and time, and want to combine or integrate multimodal data. With one of the key components in the Human Brain Project is to utilize a very broad toolbox for the most advanced information and communication techniques to help the decoding the human brain. And well, from the development of microscope imagery technique, researchers in neuroscience want to develop a brain connectome atlas. And actually, the challenge is that we uh, can obtain your ultra-scale data volumes from the optical microscopic devices or electronic microscopic devices, and we usually get very huge data volume. For example, even for a fly brain in size of 1 cubic millimeters, and it costs about 16 months to capture the EM data in the physical resolution of 4 by 4 and 40 nanometers. And the data volume is about, 106 terabytes. And now for uh, a mouse brain, we cannot actually, it is impossible to capture the high resolution microscopic images for the whole brain, which in size of 1 cubic centimeters, and it's even impossible for capture the whole data of the data of the whole human brain. So we typically get only a very small part of the mouse brain or human brain. But even though we actually have petabyte scale data volume, and in order to efficiently analyze all this microscopic image data, so researchers have the high demand on efficient data analysis tools. So it brings the opportunity for computer guys to develop computer-assisted technology to help neuroscientists to build to analyze the microscopic images, to reconstruct the 3D neurons, to segment, and to do morphology analysis. Well, due to the very super high resolution of the image typically the resolution is nanometer, but a neuron, even a single neuron, it covers almost the whole brain or even, a half a brain. So it's very difficult to trace and to reconstruct the whole complete 3D model of a single neurons in the whole brain. Here we have some examples of how detailed or how fine scale is the neuron structures. And the left figure is a cubic millimeter sized chunk of a piece of a mouse cortex. And the right figure shows only a few of the reconstructed mouse neurons. And here is some snaps dendrites with the vesicles, which describe how the neurons interact with each other, how the signals transmitted through different neurons. Well, the vesicles is shown in the white dots here. So we can see that, even to build, it's very, very challenging to build a whole brain connectome atlas. For there are a lot of research groups in all over the world. For example, the Leachman's lab is a very famous neuroscience lab in Harvard University. They develop a lot of computer graphics tools and visualized tools to facilitate the construction of a Brainnetome atlas. Well, it starts from the sliced images in very high resolution using serial multi-sense electronic microscopic devices. And then they use some image processing techniques to register the image slice together and build a 3D volume data of the piece of the brain tube. And then we typically under the physical resolution of a nanometer scale, we usually get a petascale volume of the data. In order to extract the neurons, different cells, and to analyze the structures and the functional of the neurons. So we typically need the petascale volume rendering of the underlying microscopic data, and we also have to do automatic segmentation. And even with the well-established segmentation techniques, it still requires a lot of human proofreading work to connect the over-segmented pieces together. Then we can do the morphology analysis and the visualization of the neural structures and similarities related to functions. Researchers have devoted a lot of efforts to develop tools, computer techniques to help neuroscientists to do data analysis in the petascale image analysis. And we started our work on Brainnetome Atlas construction since 2016, actually. And we worked together with the scientists in our in the School of Biology, University of Science and Technology of China. And actually, they provide a whole brain of optical microscopic images of a mouse, and they just want to find someone who can develop computer automatic data analysis tools to actually to reconstruct all the neurons in the whole brain, and then they want to do some the whole brain reconstruction of neuron populations. Then they want to do morphology analysis of the neurons because the structures, the shapes, and topologies are very highly related to the neural functions. So based on this rough idea, we have conducted extensive research on studying new techniques for data analysis or 3D modeling of neurons in the whole brain microscopic images. And today I will talk about the three parts of our recent work. Well, the first part is that to reconstruct 3D neurons from the volumetric data. Well, though we have witnessed the fast development of the 2D semantic segmentation algorithms and 3D point cloud segmentation algorithms in computer vision, in computer graphics. But for data analysis or segmentation or 3D reconstruction in neuroscience, we face a lot of new challenges because of the data has different characteristics compared with the nature image, and we also have some new challenges on data annotation. You can see that the image, we usually have the black and white, the grayscale images, and we have a huge data volume, and the image quality is quite actually is not high compared to nature image. And also we need to do very dense 3D reconstruction segmentation for the fine scale structures in nanometer scale. And also for a complete single neuron, it covers a very significant long range across the brain compared to the physical resolution of each voxel. So the traditional reconstruction method usually requires a lot of parameter tuning work because there are different devices. The image quality varies a lot, and the traditional methods are usually very not robust enough, and also because of the parameter tuning work, and also the long range of a whole neuron structure, so they typically require manually tracing of the fine neuron structure. And if we want to apply deep learning techniques on this particular application, well, we all know that for these deep learning methods, we usually require sufficient training data, but in this particular area, the data annotation cost is very high, and because we cannot use crowdsourcing, we specific domain knowledge is required to do data annotation, and we don't have enough training data, and so usually the model trained on neural net on nature images is poorly generalized to the macroscopic images in neuroscience. And well, in order to eliminate the requirement on annotation data, we want to apply some high-level pliers to help the efficiency to help to improve the efficiency on deep learning based algorithms. Well, especially we are working on this 3D reconstruction and the morphology analysis for neurons. We want to encode the structure priors of the neurons to develop deep learning techniques and to in order to reduce the data annotations. Well, we can see that in order to generate the training data with annotation, we can actually use the traditional method to generate some pseudo labels to for data for network training, but the pseudo labels usually contains a lot of noise. So we want to use the structure prior to guide the training of deep neural networks to elevate the impact of noises. And based on the idea, well, I will introduce some specific techniques applied for neuron reconstruction and the segmentation and the morphological representation learning. Where the first work is neuron reconstruction from OM image, which means the optical microscope images. And the task is defined as given data volume, a volumetric data set, and we want to reconstruct all the three neurons with skeletons. And so, well, the challenge includes, well, the deficiency of data annotations, and they have a large variety of complex neuromorphology, and actually the image quality is very low compared with the nature images. So, well, there are a series of traditional methods for neural tracing with handcrafted features, but actually the traditional methods are very sensitive to image noises and require parameter tuning. And since 2016, there are some studies to apply deep learning, especially for 3D segmentation networks on the macroscopic image, and actually they spend a lot of efforts on data annotations. So we want to improve the segmentation and reconstruction performance without requiring more data annotation. And the idea is that we can use some traditional methods to generate the pseudo labels for data network training, but then we can progressively update the network and update the tracing results to iteratively improve the power of the segmentation network. So usually, if we want to do neural reconstruction from a volumetric OM image, we first segment the signals of the neurons first, then do some global tracing of the skeletons of each neuron. But in order to train the 3D segmentation network, we need the volumetric and the both voxel-wise label of the neural signals. But actually, due to the super high resolution of the images, we actually it require a lot of manual efforts if we want to generate the training labels, the segmentation labels for training the segmentation network. So in order to generate the data annotation manually, we use some traditional neural tracer. Well, the neural tracer actually implicitly applies some high-level or more global priors on the neural structure. So we start from the OM images and we apply some traditional tracer to generate neural models. So because of this traditional tracer might be very sensitive to image quality and to image noises, so the pseudo labels are not perfect enough, but at the first iteration, we can train the segmentation network, then generate a segmentation model. Then we use the segmentation results to enhance the original image. Then we fit the enhanced images into the neural tracer again to generate better reconstruction results. Then we iteratively train our segmentation network and generate better reconstruction results. So with this progressive learning framework, the segmentation network becomes powerful and more powerful and powerful. So after 3 to 5 iterations, we can improve the segmentation network a lot. So this shows the results of our progressive learning neuron population reconstruction results. We first compare the performance of a single neuron reconstruction on the bigger neuron dataset. So you can see that compared to the manual tracing results, our results can generate very complete neural structures. And here's another results. And our work can also work on the 3D tracing for multiple neurons together where we build our data set called Visor 40. And so compared with the previous traditional tracing method, ours have more complete neural structures. And we also show some quantitative comparison, and you can see that with different traditional tracers, our progressive learning framework can increase the reconstruction accuracy. But you can see that for this framework, there are still a lot of noises in the pseudo labels, even we get better segmentation results and better tracing results. And also, it requires several iterations for progressive learning, and for each iteration takes a lot of time and the computational resources. So we want to remove the noises in pseudolabels with higher level structure priors, we found that actually there are a large number of reconstructed neurons in the public dataset, but there is no original microscope data. So we cannot use this for pairwise training, but actually these reconstructed neural models provide us the high-level morphological priors. Well, how to represent these priors? We propose to encode the morphological priors with a GAN framework. So the idea is, well, we can train a segmentation network with the pseudolabels generated by traditional neural tracer, but in order to evaluate the segmentation results, we not only use the voxel-wise loss for the segmentation, but we also measure the capability of the goodness of the reconstructed structure according to the morphological prior learned or encoded with this GAN framework. So according to, even we don't have the original microscopic data in the data set, we crop the 3D, the part of the reconstructed neuron structures, and then we use a discriminator to evaluate the goodness of the segmented structure. So with this generative adversarial network framework, we can train the 3D segmentation network with noisy labels, but with the guidance of high-level morphological priors, we can improve the quality of the segmentation results. So this framework does not require iterative training. It only requires one-pass training with the existing 3D models for morphological priors. So we compare this we name our network as MP Neuron Reconstruction then. So compared with different traditional neural tracers, our method can significantly improve the completeness of the reconstructed neural structure. And compared with our progressive learning framework, this MP again achieves comparable reconstruction accuracy, but it significantly reduces the computational and memory cost since only one-pass training is required. And we also show some reconstructed results here. You can see that compared to the ground truth label, our method can generate a more complete structure for the long neurons here. And then since due to the limitation of the hardware or computation or memory storage resources, and we cannot complete this reconstruction, the neuron reconstruction of the whole brain at once. So we should we design a fully automatic 3D neural reconstruction system for the ultra-scale microscopic images. And well, the idea is very simple, and the key is to do neural fusion across overlapping blocks. Actually, for each brain slice, we first do 3D neural reconstruction in each block, but we actually design some propagation strategy to select to start from the cell bodies and then trace the neurons to adjacent blocks. And after we reconstruct neuron pieces in each block, we design some neural fusion methods to combine all the reconstructed the neural skeleton together to complete the neuron population reconstruction on the brain-wide size. So here we show the reconstruction results on the 50 or 42 slices of a mouse neuron, and each neuron, different neurons are shown in different color, and you can see, well, the neurons are actually, well, very different in the 3D morphology and the structures. And then after we reconstruct, well, after we reconstruct thousands and tens of thousands and even millions of neurons, and we want to do morphological analysis for the data analysis to understand the relations between neural structures, morphology, and functions. So we should develop some efficient or very powerful morphological representation to for large scale data analysis. And based on a powerful morphological representation, we can do neuron classification and we can do neuron retrieval in a large scale data set. Well, but the difficulty is, the challenge is, well, how should we describe the morphology difference or morphology features of a neuron according to the 3D skeleton or 3D surfaces, right? So, well, because of the reconstructed neural model have naturally the connections between different branches, we propose to use the graph representation and apply. We want to learn the morphology representation based on graph neural network. Well, also we face the same challenges that we don't have enough training data, and so we want to apply contrastive learning techniques to do unsupervised training with a large-scale neural database. So we design some neural augmentation methods, including the branch dropping, the pointer propagation methods to generate different copies of one encoding neuron. And then we design a morphology graph neural network to learn the morphological representation of each neuron. So by constructive laws, we can train the morphological graph neural network to learn the embedding for the encoding neuron based on the graph representation. And then for large-scale neuron retrieval, which has very high requirements on the efficiency, we want to further convert the learned embedding vector, which are continuous values, into a binary hash code. So we design a second part of the morphological representation network called the binary hash code embedding network for high efficiency. But the difficulty is, how can we train this binary code based on together with the neural graph neural network for morphological representation? Well, we design a alpha blending strategy based on the straight-through estimate. So we all know that in order to train the feature embedding part, well, for the binary hash code, we actually do the gradient estimation according to the continuous value, but actually we do a fusion by increase the weight for the binary and the continuous embedding value z to progressively train the feature embedding network. So after well jointly train the graph neural network for feature representation and the binary hashing code embedding network together. We improve the representation of the capability of the graph neural network. Here we show the results on a neuron classification based on a database that contains 5,000 neurons. Well, these are the two variants of our model. One is just with the embedding Z, which have the continuous value because and the hashing map CNN is generates the binary code. Well, we can see that well with the more precise feature representation, the MACGN achieves the highest performance on neuron classification. But even with our binary hashing embedding, we can obtain very similar results with the map GNN. And we do a set of ablation studies on the neuron classification network. We can see that with the full model, and they compared with the full model, the hashing can actually generates very comparable results for each type of neurons. And also, well, because of the hashing binary code is particularly designed for fast neural retrieval on a large scale data set. So we compare the retrieval performance on two data sets, the 5K 1,000 data set and the 100,000 data set. We can have very similar performance, but with the binary hashing code, the retrieval speed is very faster than the original MapGNN. And here we show some retrieval results. We can see that we're based on our morphology representation, we can very efficiently get the neurons have similar morphology and similar shapes. And according to the manual label, we can see that the reconstructed results is assigned as the same type of neurons. And then, well, this is designed for more complete neuron skeletons. And actually, there is another type of Marcus Ford images, which in the resolution of a nanometer. Well, we see a lot of techniques have been developed for automatic segmentation on this volumetric EM data, and typically they generate over-segmented results. And in order to get the a complete neurons, well, there's a paper published by the Cern's lab in Princeton. They reported that, well, in order to reconstruct or to trace the about 1,000 neurons, it costs about 50% a year of human proofreading, given the automatic reconstruction results. So we want to do some well, data selection or data filtering for the whole brain neural cases. So we want to classify the well, the over-segmented pieces according to the 3D shape, and then we design another unsupervised morphological representation for EM segments, where we represent the neuron pieces based on the surface points, where it works like to learn a morphology representation for 3D point cloud, where we use the same idea of contrastive learning. We follow a typically contrastive learning framework, which requires the data augmentation method to generate the two contrasts of the input, and we learn the representation, projection, and compare the prediction between the different views of the same input. But actually, the instance level contrast have difficulties to represent fine scale difference of the 3D shape. So we design a cluster instance contrast model to enforce the representation learning to make the distribution more compact for the same cluster. So we design a clustering strategy and compare the predicted pseudo labels and the cluster instance similarity to make to enforce the compactness of representation learning. And we compare our method with some typical state-of-the-art contrastive learning method, including the SWAP and the BYOL, and we can see that our method achieves the best results on the neural segment classification. And we also visualize the learned morphological representation in the feature space. We can see that without the sub-feature B is the results without our cluster and instance contrast model, and with our cluster instance contrast model, we can the feature embedding are more discriminative for segment classification. So in conclusion, we can see that the computer graphics and computer vision techniques significantly facilitate the ultra-scale data analysis for the brain connectome atlas reconstruction. And in particular, we focus on the 3D reconstruction and morphological analysis of neural models, and we apply some structure-guided idea for semi-supervised learning or unsupervised learning to exploit the high-level structure priors and reduce the requirement on data annotation. And actually, we can see that, well, based on this whole workflow for connectome reconstruction, we still have a lot of opportunities for data analysis and visualization, and especially for the long range neural tracing, for the visualization, and also the simulation technique is also desired to simulate the dynamic neural activities and recover the relation between neuron structure and functions. So this is my talk, and you're welcome to ask questions. Thank you.
Okay, thank you very much. Thanks for Chen's great talk. So it is very insightful, and it's very interesting to see that there are a lot of quite general methods that it can also be utilized in different kinds of quite specialized medical data. So while the team is collecting questions, I would like to start with my own questions for you. So I would like to ask several quite general questions. So for your 3D and 2D neural reconstruction work, actually, especially for the 3D reconstruction, how the success of the 3D neural reconstruction measured? I saw some slices for the 2D things. So are they the thing to measure, quantitatively measure the success, or there is some other specific way to measure the success of the reconstruction?
Okay. Do you mean the images like this one.
I mean the 3D neuron reconstruction one. Yeah, I believe it's this one. Several, tens of slices. Yeah.
Oh yeah. Actually, well, this is actually a cube. This is in 3D, but it looks like 2D because we project the pixel intensity onto the 2D plane. But actually, well, yeah, this one is actually a cube. So and the reconstructed results are also 3D skeletons, but let me show you the. I think I have a visualization. Oh yeah, here one. You can see that actually it's very a thin slice, but it's in 3D.
Oh, okay. So they are not a 3D volume by accumulating 2D layers. It is a real 3D volume.
Yes.
Okay. Okay, thanks. Yeah. So I also had a question about all the deep models you used for reconstructing the neurons and doing the analysis. So there are a lot of pre-trained models for segmentation, for reconstruction, for even for GANs, for image processing. So how generalizable of these models in your specific area of those medical image processing? I mean, are you using all the models? Are you training all the models from scratch, or you can also transfer some pre-trained models and to get a better results?
Yes, we take the common structure of a segmentation network like UNet, 3D UNet, but we have to retrain it with from random initialization because the pre-trained model using the nature image doesn't work on the macroscopic image because you can see that where the image, the textures and the intensity are very different from what we have for nature objects. So we really retrain our segmentation network from scratch.
Okay. Yeah. So all right. So actually yeah, yes, because your works are actually very interesting. So I have another question for you. So for the morphological operation, morphological analysis on the I believe it's the neuron structure. You have a graphical model there. So I wonder, yeah, so you have you build a graph here. So I wonder for those microscopy images for those neurons, is a structure similar for all the different neuron images? So that you can give a fixed kind of a structure, and you just need to figure out the difference between their lengths and nodes, or the topology of themselves, of, for example, the node numbers and how they are connected, are the distinguishable features you are going to analyze?
Yeah, very good question. Actually, well, the traditional method in this area with the neuroscientists usually define some rules or use some handcrafted features to describe the topology, like what you say, the node number, the branch numbers, and the angles between different branches, right? And these handcrafted features cannot be well generalized uh, to more types of neurons because with the automatic segmentation method we get more and more neural data. And with this handcrafted feature, it's very difficult to find the fine scale difference between the neuron structures.
Okay. Okay. So yeah, let's thank Professor Chen again. Thank you for your excellent report.